Welcome to the NIIMBL American Rescue Plan webinar series. This webinar series highlights the projects funded through the American Rescue Plan. The goal of these projects is to help the nation prevent, prepare for, and respond to coronavirus public health threats. In this webinar, we will hear from Abraham Lenhoff and Wilfred Chen from the University of Delaware on the project Affinity Precipitation Purification of virus-like particles. You can find additional webinars in this series on our website, www.niimbl.org. And now we will turn it over to Bramy.
Thanks very much for the kind introduction, Dan. We're especially grateful to have had the opportunity to work on this interesting project, and we'd like to thank NIIMBL for the support and thank the ARP program. Our project involves affinity precipitation purification of virus-like particles, and I'll start off by providing a little bit of background on the project. These virus-like particles can be used as vaccines and have been used in vaccines for quite a long time, but they're now finding increasing use also in gene and cell therapies. And what makes them interesting is that if you look at the panel on the right-hand side, is that even within the space of biologics, these VLPs are really big and complex moieties to deal with. What's shown here are the sizes of different drugs. This is obviously aspirin, a small molecule drug. These are the low end and the high end of the biologics, the typical biologic spectrum, and insulin and a monoclonal antibody. But even for a relatively large biologic drug, something like an adeno-associated viral vector, which is actually a relatively small virus-like particle, these things are very big. And what that means is that their size and complexity can make manufacturing expensive and inefficient. And that's really where our project is headed and what it's directed at. The reasons for the complexity of VLP manufacture are that the fermentation itself, growing these VLPs in living cells, can be very expensive, even for a relatively small fermentation, such as a 100-liter one. Purifying these adds an additional complication, and typically your yields can be very low. This is below 50%, which is really intolerably low even for a biologic. And so what all this means is that you need to do a lot of work to scale the project up in order to make enough VLP suitable for use in patients. One of the main reasons for the complexity of VLP manufacture is a particular set of steps called chromatography. And this is where the VLPs differ even from relatively large biologics. In chromatography, what you're trying to do is to purify the target molecule by binding it to a surface. And in order to have enough surface area to bind to, what's typically used are porous particles, such as what's shown here. And so what you can see is schematically, we've put a lot of these molecules inside the particle. When you're dealing with a VLP, it is so big that it can't get into the particle. And as a result, you've got room to stick to the surface only on the outside surface. And so your utilization of your available surface area is very low. And so this is the reason for looking for alternatives. And that's where our project came in. And so I'll hand it over now to Wilfred to explain that context.
Okay, so what we wanted to do in this project is to go away from using chromatography, but take advantage of precipitation. Okay, but unlike conventional precipitation, we wanted to be able to do a very specific capture and release mechanism. Okay, so this process is typically known as affinity precipitation. So the way we wanted to do it is we will insert a binding ligand onto the VLP of interest, and then we'll synthesize a capturing moieties that can bind very specifically to the binding partner on a VLP. Okay, and upon binding, we'll find a very simple way to allow the capture VLP to form a very large aggregates. Okay. And so the outcome is they will instantaneously precipitate and crush out solution. Okay, which make it very simple in terms of the purification from the rest of the contaminating components, particularly like proteins or other proteins in the media. Okay, and once we recover and capture the VLP, we will then use a second step to break the interaction, and therefore the VLP are now released back into the soluble solution, and then we'll induce again the aggregation of the capturing ligands, so we can actually now remove in the final step and recover the VLP of interest. Okay, so the framework itself is actually quite simple as captured in the cartoon in this slide. Next slide, please, Amy. Right, so the question is, how do we actually enable the specific interaction? And also, how do we actually induce spontaneous aggregation and precipitation? So there are a couple of different ways for us to do it. To identify the particular interaction domain is actually quite easy. There are a variety of different ways to go by doing it. To induce aggregations, we actually investigated two different types of strategies. The first strategy is to make use of a very simple protein called elastin-like polypeptide, or ELP, which has the capability to go through a reversible phase transition. At room temperature and low salt concentration, it's essentially completely water soluble. But when you raise the temperature to like 30 and modulate the salt concentration, the ELP will spontaneously aggregate and crush out solution. So that's one particular mechanism that we can explore to enable the phase transition and aggregation to occur. The second major mechanism that we actually use to capture the VLP is based on multivalent interaction. Okay, so you can imagine that in a single ELP, there are multiple different monomers, in some cases up to about 100, 200 different copies. If every single one of them is capable of interacting with the capture ligand, when the binding occurs, it will spontaneously cross-link, illustrated by the picture on the right-hand side, okay, based on a multivalent interaction. Okay, so all the VLP now form a massive cross-link network. Okay, and when the size becoming microns or larger microns in sizes, they will cross out a solution, and you can easily use either filtration technique or centrifugation to remove it from the soluble solution. Okay, so this is the two major strategies that we use in the proposal and in the work to enable us to do the binding and also the spontaneous cross-linking and precipitation steps. Next slide, please, Amy. So I think the animation in the movie we're showing you on the right-hand side has a really good representation of what the process looked like. So initially, we have a protein solution we want to capture. When we add the capture ligands into the solution, not only would it bind to the intended target of interest, but also induce a spontaneous aggregation, and therefore you can see the solution become very turbid and cloudy. Okay, so this is the kind of framework that we use for both purifying smaller protein, but more importantly, the VLP particles of interest in our work. Okay, so what we have been able to accomplish so far is genetically able to produce the capture ligands with decent and reasonable productivities. We're able to show that it can bind very specifically to the target VLP of interest. It can also induce spontaneous cross-linking and precipitation without any other salt. Okay, and as a result, we were able to recover close to 90-some plus percentage of the VLP particle directly from the cell lysate, and we're able to use the elution step, washing step, to ultimately recover more than 90% of the VLP that were produced in the original cell culture. Okay, so but more importantly, we also show that this process can be reused, which means the capture ligand, when we release it from the binding, we can use it again for a subsequent step and an additional VLP purification. Okay, and by working with when a postdoc and the student working with Professor Lenhoff's group, they were also able to now show not only can we do it in a batch process, We can also use it in a flow system, which Brandon is going to talk a little bit more about it.
So the last part of the project involved taking things from the lab scale to something a little bit closer to at least the concept of larger scale manufacturing. So what we're showing here is really a series of still pictures showing the individual steps in doing the precipitation, the washing, and then the breaking up the complex and recovering the product and the ligand for use, the ligand for reuse, and the final product, obviously, for use as the product. As you can tell, doing this in multiple individual steps can be quite inefficient. We don't have scale bars on this, but these vials are really very small. And so the volumes that are being used here are very small. So this really isn't suitable at this in this form for a large scale separation. So what we did was to demonstrate proof of principle for use in a continuous process. And in a continuous process, you would be feeding your reagents in continuously, shown here on the left-hand side, as well as some additional reagents along the way. And you'd have continuous flow through the system. There'd be different steps happening along the way, depending on which reagents you added at different steps along the way. And what you get out at the end would be one stream containing your product, one stream containing the ligand, and you'd be able to do this in continuous fashion. Running something like this in continuous fashion requires enormous amounts of reagent, and so we weren't able to do that. But what we were able to do was to demonstrate the principle of running each one of these steps individually. And so we think we've established the proof of principle for this process for potential use in actual manufacturing. And so just to summarize, what we've done has been to design and synthesize the whole molecular machinery for doing this kind of affinity precipitation on virus-like particles. We've demonstrated the full capture and recovery cycle, and as Wilfred said, with 90% plus recoveries on the lab scale, and we feel that we've got all the components and the infrastructure and the demonstrated performance of the individual steps for potential use in a continuous operation. So that's the project in a nutshell, and we'd be happy to answer any questions.
All right, thank you very much, Bramy and Wilfred, for the great presentation. First question, anything in particular surprise you, or any challenges that you had to overcome during the project?
I would say that the biggest challenge was actually getting the engine actual virus particle, the adenovirus particle. So we actually tried and worked with a company for over 6 months, and they were not able to produce the required viral particle that has the affinity partner. Fortunately, we're able to substitute using a different VLP particle, roughly about the same size. So we're able to use the surrogate and demonstrated the concept works. We're consistently getting over 90% recovery. So that was challenging for about 6 months, but fortunately, by working with the NIST people, we're able to find a suitable substitute to still demonstrate the concept.
And the irony of that, of course, is that we experienced firsthand the difficulty of making these virus-like particles, which is the whole object of the exercise.
Excellent. And the second question, what's next for the technology? Is it safe to presume maybe it's continuous testing of the continuous methods that you've been able to get a start on?
Yeah, I think actually running the full continuous process would be great to be able to do. That would require, as I said before, large amounts of material, and we just don't have those large amounts. So we'd have to find a way of making those. But there are biopharma companies that are looking at this kind of continuous flow technology, whether it's affinity precipitation or regular precipitation, in trying to operate that continuously. And there are also a couple of small companies that are working on related technologies, not necessarily for VLPs, but this kind of affinity precipitation process again, for manufacturing purposes.
Great. Well, thank you both for the fantastic presentation, and thank you for watching our American Rescue Plan webinar series. To find additional webinars, visit www.niimble.org.